Hi everyone. So we are group 5 and the article we chose to present on is cell-to-cell movement of mitochondria in plants. So today we will be analyzing the effects that are created after transferring DNA from one species to another. We will discuss the hypothesis, the materials and methods used to carry out the experiment, and go over the final results. So, to fully understand why and how the experiment is going to be done, we will first go over some key elements, which include defining mitochondria and horizontal gene transfer. Mitochondria are very important organelles in a cell, which is why they are normally referred to as the powerhouse of cells. They are involved in several cellular processes, which include ATP energy production, calcium homeostasis, and the regulation of reactive oxygen species generation. Chloroplasts will sometimes function along with the mitochondria in certain metabolic pathways to produce an efficient exchange of metabolites. Mitochondria are able to move at various speeds while in plants, which depends on the cell type and species. In tobacco cells, the maximum speed that the mitochondria are capable of moving at is 2.5 to 10 micrometers per second. It has been previously noted that chloroplasts may be able to influence mitochondrial movement, but not proven yet. So, whether we have two organisms of the same species or of different species, we are still able to transfer genetic material from one of them to the other. This can be done by using a method called horizontal gene transfer. Horizontal gene transfer is when genetic information can be transferred from one organism to another without it being its offspring. In plants, mitochondrial DNA is usually involved for a gene transfer between the donor and its recipient. One mechanism of horizontal gene transfer was found to be grafting. It was discovered that specifically tobacco graft junctions were able to exchange plant DNA and create a movement of the chloroplast genome. Although this means there is cell to cell movement of organelles, there is still no evidence of cell to cell movement of the mitochondrial DNA. It can now be understood that the main purpose of this article is to find evidence of mitochondrial movement in plants. A horizontal gene transfer event was first shown between the two species N. undulata and N. tabacum. A cytoplasm from one of the flowers was replaced with the cytoplasm of the other flower, which ultimately produced a conversion of anthers into stigmatoid petals. Because of this, the experiment can be performed expecting that anther morphology should be restored because of the movement of N. sylvestris mitochondria in CMS cells. Although cell-to-cell movement is the main purpose of this article, it is important to ask whether mitochondrial movement will produce any changes. Specifically, will cell-to-cell movement enable a modification of the mitochondrial genome by DNA transmitted from two species? It is also important to determine what the effects of the modifications will create. Would a change in the mitochondrial genome lead to any significant changes, and how does this ultimately help scientists in the future? So, to actually carry out this experiment, a hypothesis needed to be formed so it could be tested using research. The hypothesis that was created for this experiment is graft junctions regenerate plants to restore fertile flower anatomy for mitochondrial DNA transfer. To test this hypothesis, plastid graft transmission events will be performed, leading to a regeneration of the plants. These graft junctions should also be able to show a recombination of the mitochondrial genomes. The hypothesis can be determined as false if there is no evidence that the anther morphology was restored after a mitochondrial DNA transfer between N. sylvestris, and N. tabacum.
Today, we'll be discussing the materials and methods that were used in this article. And we'll begin with talking about the graph partners that were used. We begin with the Nicotiana tabacum line that carries cytoplasm of the Nicotiana undulata, and then that is then transformed with the agrobacterium binary vector. This would then yield the gentamicin resistant line NTG115 and also the NSF, which is the fertile Nicotiana sylvestris line. Plastids from this line were then plastids from this line were then transformed with the plasmid PCK2, which were then encoded spectinomycin resistance along with the visual aura biolophysose resistance genes. The chromosome identification by simple would be the next method used to create some of these figures that we've seen in the results. The PCR program that was used in this part, we'll go ahead and discuss that now. Settled trimethylammonium bromide was used to isolate the cellular DNA, and this helped create the simple sequence repeat markers necessary to go ahead and begin the PCR program. And began with the first cycle, which was ran at 94 degrees Celsius for 5 minutes. This is the part of the denaturing process, which splits the DNA strands in two. And this was ran for 37 cycles at 94 degrees Celsius for 45 seconds each cycle. And then the next part of the PCR program involved it being ran at 59 degrees Celsius for 45 seconds. And this kind of allows the nucleotides to be added. Part of the cycle begins with we'll run at 72 degrees Celsius for 1 minute. And 72 degrees Celsius for 10 minutes was used for all of the chromosomes, except the ones that were 8, 12, 14, and 16. These chromosomes went under a different PCR program, and that then consisted of 94 degrees Celsius for about 5 minutes, which again is the denaturing process. And then 37 cycles of 94 degrees Celsius for 20 seconds apiece, and then 54 degrees Celsius for 20 seconds, and 72 degrees Celsius for 1 minute to allow the annealing process to begin. The third method used in this experiment was the restriction fragment analysis of ptDNA, which is the plastid DNA. And this DNA was then purified with Seattle trimethylammonium bromide, which is then digest the BAM-HI restriction enzyme, and that is then probed with the RRN16, AADA, and bar probes. That one is one of the more simple processes. It just only takes the one step. The next generation sequencing is the next method we'll be discussing. Next generation sequencing involves determining genome sequences. The Seattle trimethylammonium bromide purified DNA physically shears with the Covaris system. And the sequencing libraries created from this were then prepared with a standard true sequencing DNA library preparation kit, which is just one of those kits you can buy off a scientific website, which allows you to, which helps you go along with the next generation sequencing program. These libraries were then selected by size at 650 base pairs. This was done using the Eagle Agros electrophoresis system, which is another piece of scientific programming that they bought that you can use online. You can get it yourself. And it was then quantified with the Qubit DS DNA high sensitivity kit, which is then another one of those scientific programs that they ordered to allow them to continue on with this experiment. And for more on next generation sequencing, the size of these fragments were analyzed using bioanalyzer from Agilent Technologies, and that's the ones that were selected at 650 base pairs. The normalization and sequencing of these fragments using MySec V3 chemistries, 2x300 base pairs, and the trim adapters and barcodes received from this were analyzed in a default setting in the Illumina experiment manager.
So on the next slide, we're going to be talking about plasmid genome assembly. But before that, I think it's important to give at least a little background on it, as it's probably a new subject to a lot of us. So as the name implies, the process is assembling the nucleotides into its correct sequence. This is done by the use of a genome assembly program, and that fragments the that puts together the fragmented DNA into a whole, so that we can read. So the authors of this study use the Burroughs-Wheeler alignment maximal exact matches, and they use that due to it being fast and accurate, as opposed to the secondary option, which was the angular effect, which I guess due to what the authors were researching did not deem it effective enough for their study. And this was also they also used the de novo reads. De novo is Latin for from the beginning or anew. So this is used when there is no reference sequence available to us. So they're just starting fresh, making it new. And the assembly requires the use of memory intensive programs, more so with the de novo. So with the background we have now, the GT19C plasmid is assembled. And this is done with the use of adapter-free quality trim reads from 4 GT19C offspring, which were mapped using the same program mentioned before, the Burroughs-Wheeler alignment maximal exact matches algorithm. And this was used for creating the de novo contigs using the Abyssal system, the program. And again, the de novo means new. And this was paired with the option with the K-mer of 90. Okay, so this was used for creating the de novo contigs using the Abyss program. And again, de novo meaning to begin new, since they don't have a reference point, which is why they're using the program. And the settings for this included the paired in option with K-mer of 90, which went off the weight.
So it was paired with the end option of the K-mer with K-mer at 90. The infectious salmon anemia virus, segment 5 putative acetylrase P3 gene was used as a guide to map the contigs. And there was 4 identical DNA sequences of offspring that were produced. And the DNA program used to compare these DNA sequences was MVista. And they also used the N. sylvestris plastid DNA, and it involved a 300 base pair sliding window. And they used the Nicotiana undulata markers, and those are blue, as you can see in the picture, and then the L. sylvestris markers are in red, as seen in the picture. And this was the what they got. This is the results of the GT19C plastid genome assembly. The mitochondria DNA mapping and contig assembly was another one of the methods that were used in this article. They used the adapter-free quality trim reads, and these are mapped with the mitochondria DNA of the Nicotiana tabacum, Hyoscyamus niger, and 2 Capsicum unknown cultivars mtDNAs using the BWA-MEM method that was stated previously. And the maps of this reads, along with their pairs that were created in the de novo contigs used in the last step. And the meaning of these sequences were a genome where no reference sequence for alignment is available. The Nicotiana undulata mitochondria single nucleotide polymorphisms were identified by GATK haplotype caller program. And these single nucleotide polymorphism had an 80% ratio, greater than or equal to 80% ratio, and the minimum 30 coverage in Nicotiana undulata. The origin of the regions and seed progeny were assigned by these single nucleotide polymorphism and de novo contigs. So when you see them line up against each other, they're categorized by the number of polymorphisms they had in each of the contigs.
So again, I'd like to give a little bit of context to what the next part of the experiment pertains to, and that is RNA gel blot analysis, probably better known as northern blotting. And this is very similar to another process you might have encountered in one of your first biology classes, which is southern blotting. However, in northern blotting, it runs opposite. So RNA gel blots is a procedure, and it is used in gene expression by detecting the RNA. And the RNA first needs to be extracted, and it needs to be separated from the okay, so procedure begins by extracting the RNA from the homogenized tissue and cells, and that is first done by soaking it in a solution which separates the two, and then it must be um, spun in a centrifuge to separate it. And once that is done, it is put into separate wells, and by process of gel electrophoresis, it is separated by its molecular size. And the matrix of the gel works in such that the larger it is, the slower it runs, and the smaller it is, the faster it runs. So when it's complete, um, it is run by a probe complementary to the target sequence. And the probe is already a size that is known. So when that probe is analyzed to see where the middle wells link up to, that's when we know that's the probe complementary to it. RNA samples are then transferred to a nylon membrane with a positive charge, and there's weight on top of it with paper towels and a capillary blotting paper. And what the weight does is separate it from the bottom with the transfer buffer, and it rises up from the agarose gel to the membrane. From there, it is heated with the UV or a UV light or just heat, and that immobilizes it through covalent links. And once that's done, we put it through a process known as dyssynthontry. And this X-ray film is able to be read through a image processing software, and now tells us exactly where it links up on the gels. So RNA gel blot analysis used for this specific example, for this study, used Trizol, which isolated the total cellular RNA from leaves of the greenhouse grown plants and dissolved in 20 microliters of cyclocarbonate water. RNA precipitated by adding 2 microliters of 3 molar sodium acetate and 66 microliters of 100% ethanol. RNA is then sedimented by centrifugation, washed with 75% ethanol, air dried, and dissolved in 22 microliters of DEPC water. 3 micrograms of RNA was electrophoresed in 1.5% agarose formaldehyde gel in MOPS buffer. PCR fragments used as probes and were amplified using a total cellular DNA as a template. And dead RNA gets gel blots were hybridized with ORF293 and ATP1 probes. Seed progeny of CT19C marked by adding 1 more digit for each generation separated by a hyphen, which you can see here on the figure on the right.
So next, I'm going to be going over some of the results, which include plastid graft transmission events, recombination of mitochondrial DNA, as well as the expression of ORF293, which correlates with homeotic conversion of anthers. So a bit of background information. The CMS N-tobacum graft partner, labeled as NTCMS, carried a nuclear genomycin resistance gene. Fertile N. sylvestris, labeled as NSF, graft partner carried 2 plastid markers. The first was selectable spectinomycin resistance aminoglycoside 3 adenyl transferase, also known as AADA gene. And the second was visual Aurelia bialophus resistance leaf color gene, which is depicted by figure 1A. 2 Nicotiana species grafted are depicted by figure 1B. They were sliced, and resistant shoots from these tissue slices were selected in culture specifically for genomycin resistance. The resistance is encoded in the nucleus of CMS N-tobacum graft partner, and spectinomycin resistance encoded in the plastids of the fertile N. sylvestris graft partner. 3 clones with resistance to antibiotics were recovered in 14 graft junctions. These were labeled as GT7, GT17, and GT19. The regenerated plants were confirmed by a DNA gel blot analysis of all 3 lines as shown in figure 1, as well as the sequencing of the genome of GT19C line. Chromosomes of N. tabacum were found to be carried solely within regenerated plants. Selection for nuclear marker in N. tabacum and chloroplast marker in N. sylvestris yielded N. tabacum plants with chloroplasts from N. sylvestris without input from N. sylvestris chromosomes. 3 double resistant cali. 8 plants regenerated, and within the 8 regenerated plants, all but one had CMS flowers. GT19-C was chimeric, which means it contained fertile flowers on 2 of 4 branches. This suggests co-transmission of fertile mitochondria with N. sylvestris chloroplasts. Double resistant plants contained co-transmission of fertile mitochondria with the N. sylvestris chloroplasts. 3 types of flowers found on GT19-C plants. The first was CMS with peletoid stigmatoid anthers, found in figure 1F. The second was fertile anthers with ample pollen in figure 1H. And lastly, an intermediate with partial conversion of anthers into petals depicted by figure 1G. The seeds of the sterile flowers were obtained by self-pollination. Seeds from fertile flowers were obtained by pollination with wild-type N. tabacum pollen. Finally, fertile or CMS phenotype of GT19-C seed progeny were obtained through 3 seed generations. Next is recombination of mitochondrial DNA. A graft junction was used to search for DNA evidence of mitochondrial movement. N. tabacum with undulata cytoplasm was selected because plastid DNA of N. tabacum and N. undulata differ by 918 ptDNA markers, and thus expect mitochondrial DNA to be comparably variable. We know that plant mitochondria undergo several repeated cycles of fusion and fission, thus the expectation is to find recombinant mitochondrial genomes. 24 polymorphic sites in 430-kb mitochondrial genome were tested, and the results were that the mtDNA in the fertile and sterile branches could not be distinguished by the 24 markers, which was depicted by figure 2. 279 kb sequence conserved between N. sylvestris and N. undulata mitochondrial genomes differ by 977 DNA polymorphic markers and encode all genes present in N. sylvestris mtDNA. Turning our attention to figure 3, the origin of mtDNA in recombinant mitochondrial genomes plotted is the N. sylvestris genetic map. Crossover sites. Crossover sites are not always identical in the recombinant mitochondrial genomes. The results were recombinant fertile 1 and 2 and recombinant sterile 3 and 4 due to the switching of the red and blue dots in figure 3. 5 recombinant junctions PCR amplified and were confirmed by Sanger sequencing. Next, I will be discussing expression of ORF293 and how it correlates with homeotic conversion of anthers. Recombination of mitochondrial genomes at alternative sites facilitated the identification of the region likely responsible for CMS, which manifests homeotic conversion of anthers into stigmatoid petals, and N. tabacum, which was found in figure 1D. ORF293 mRNA accumulates in CMS N. tabacum and sterile recombinants. 3.5-kb region correlates with male sterility. 2 recombinants with sterile flowers carried the N. undulata sequence as well as the N. sylvestris sequence, and fertile recombinants had region of N. sylvestris, which was CMS associated region from figure 3. The transmembrane domains are characteristic of sterility causing mitochondrial genes 17 and 18. Flower phenotype of plants carrying ORF in mitochondria depends on nuclear background. Finally, ORF293 was named homeotic conversion of anthers HCA gene.
And then next, we're going to go over the discussion part of this article. On the next slide, we're going to state the fate of organelles after cell movement, and it's basically going to be talking about figure 5. which is transmission and recombination of mitochondria of graft junctions. And as we can see here, the fertile N sylvestris nucleus, the red square partner, has fertile mitochondria, which are known as the red circles, and the spectinomycin resistant chloroplasts are the red ovals. And the plastids and mitochondria meet strikingly different fates. And it talks about the identification of CMS, and it also introduces the ORF293 gene. And then we discussed two ATP1 copies, which is known as the N sylvestris and the ORF293. Basically, the ATP gene preserved in both parental genome configurations. And in addition, CMS is not caused by ATP coding sequence. On the next slide, we're going to go over applications in biotechnology. Grafting enables recovery of plants, and 3 examples are going to be shown here. One is going to talk about chloroplasts of the graft without the transfer or mitochondria and nuclei. Second is going to be transfer of both chloroplasts and mitochondria in the absence of any nuclear DNA input. And third, formation of nuclear hybrids with input of all organelles. And then basically discussing the CMS transfer, graft transmission. An example that was stated in the beginning was the tobacco to the tomato. And then the ORF293 gene basically provides a visual marker useful for mitochondrial transformation. And that summarizes our article. And if you guys have any questions, please let us know. Thank you.